We left off in our introduction talking about Mendel's four hypotheses, or the four things that he sort of learned from his initial experiment with pea plants. That genes have different versions. We call those versions alleles, and that we have a genotype that actually lists out those alleles. That each organism has two alleles for every trait, one that they got from their mom, one that they got from their dad. They can be the same or different. Homozygous is the same. Heterozygous is when they're different. And then we learned that if you have both alleles, then the dominant one is the one that is expressed, while the recessive one sort of hides. And that results in the phenotype, what you actually see. That's how the purple hybrids in the F1 generation, who had a purple allele and a white allele, all looked purple, because purple is the dominant phenotype. It's the dominant allele. And so when we look, even though the genotype is one dominant and one recessive, so the genotype is heterozygous, the phenotype is actually purple, because that dominant one is going to take over and this recessive one is just going to hide. Now, it popped up in Mendel's experiments when we did a hybrid cross, because then this member of the pea plant can make big P gametes, or eggs and sperm, and little p gametes, or eggs and sperm. So it's going to give half of its offspring a big P and half of its offspring a little p. And if you cross that with another big P, little p, it's also going to be able to do that. So it's going to have a little p and a big P that it can give. And then when we look at all the possible combinations of that, the two big P's can come together, so it could have an offspring that's like that. The little p and the big P might go together, so it'll have an offspring that's like that. The two little p's might get together, and that would be that. That's the white one, because it has two recessive alleles. Or the last ones, you could have another little p, big P. So you're going to have two heterozygotes, but they're still going to be purple, and a homozygous dominant, which is also going to be purple, giving us 75%, where 3 out of 4 possibilities are purple, and just the little 25% white, 1 out of 4, turning out to be white. So then when we move on to the next slide here, we'll see that there's some outcomes of this. One of those outcomes is that the homozygous, and this should say recessive in here, a homozygous recessive phenotype, a recessive phenotype, have to be homozygous recessive in genotype. Meaning, if we see a white flower, we know that it has, and it's from the pea plant, we know that it has a little p genotype, because the only way a recessive trait is going to be shown is in the absence of a dominant. If there was even one big P and the little p, that's going to be purple. Certainly if there are two big P's, it's going to be purple. So the only way you can get that little p to show up is if it's homozygous recessive, if there are two little p's. So similarly, we see that over here in the dogs. Black is dominant to brown in labs, and so the chocolate labs are homozygous recessive. They have to have both little b's. That's the only way that you would see brown, because if there was even one big B, it would be black. Therefore, when we look at a dominant phenotype, so again, not heterozygous, but dominant phenotype. If we look at the dominant phenotype, so in this case, black dogs, we don't know the genotype of this dog. This could be a homozygous dominant dog, or it could be a heterozygous dog. Both of those would be black. So that's why it's the B blank here, because we don't know about the second one. We know it has at least one big B because it is black, but we don't know if that second B is big or if it's a little b. This leads us to an idea called a test cross, which is where we can actually determine the genotype of a dominant phenotype. So if we're looking, in this case, if we're looking at this black dog, we know it has the dominant phenotype, but we don't know its genotype. We can determine its genotype by doing a test cross, or a mating, an experiment where we mate this black dog with this homozygous recessive dog. We have to know this one's genotype in order for this to work. And the only way you know the genotype is if it's a homozygous recessive, or a recessive colored dog. So we're going to take the black dog and we're going to mate it with a brown dog. And then we look at the puppies. We look at the offspring. If the offspring are all black, then that's a pretty good indication that this dog was dominant, homozygous dominant. It had both big B's, because if that dog is homozygous dominant, the only kind of sperm or egg that it can make is a big B. So that's the only kind it makes. If it's a big B, it can make one big B or another big B. So the only kind it has is a big B. And the only one that this can give is a little b. And so if you look at the possible combinations of just big B's and just little b's, they're all going to be heterozygous, which means they're all going to look black. If this dog is a heterozygote, meaning it's the big B, little b, then half of its eggs or sperm will be big B, and the other half will have the little b. And when we breed that with a dog that just has little b's, then half of them are going to be heterozygotes, and half of them are going to be homozygous recessive, which means half of them are going to be chocolate colored. So if you do a test cross of an unknown black dog, you don't know its genotype, with a brown dog who you know its genotype because it's brown, so it has to be homozygous recessive, and you look at their puppies, if any of the puppies are chocolate, if any of them are brown, that means this dog was a heterozygote. It had to have that little b to give. If none of the dogs are brown, then that dog is very likely to be a homozygous dominant dog because it didn't have any little b's to give. So now we look into how to predict offspring. What's the easy way to do it? We haven't really organized. We've talked about, how we can combine the possible eggs and sperm that each one can give. Well, there's a way to organize that, and that's in a Punnett square. And basically what you do is you look at your parents, and you look at the possible combinations of eggs and sperm that they can give. And then you create a little chart. And the chart does not have to be a square. We just call it a square because the most common ones are square because they're 2 by 2. But they can be bigger. If you can make 3 different combinations, then it could look like this with 2 combinations. Or if you can only make 1, if like you're a big B, cross with a little b, then your Punnett square might just look like this. This one can only make big B's, and this one can only make little b's. So they can change size. They don't have to be different size. So basically, we list out all the possible eggs and sperm allele combinations, and then we make a grid where we see all the possible combinations of those eggs and sperm. So in this picture here, you have a big B, little b male, and a big B, little b female, which means this one can make big B's and little b's. This one can make big B's and little b's. And then we make the chart, we list those out on the side, and we make the chart to organize those combinations, right? So it's sort of like FOIL, where you go first, outside, inside, last. In this case, we do these 2 together would make a homozygote dominant. These 2 together would make a heterozygote. These 2 would make another heterozygote. And these 2 would make a homozygous recessive. And so that's the tool that we can use for predicting offspring. You can do this with more than one trait. You can actually trace 2 traits at a time. It takes a little more creativity. So this is actually a picture of Mendel's second experiment. He did a 2-trait experiment because he wanted to test out how the alleles were assorted. Did all the recessives always go together and all the dominants always go together? Or did they sort independently of one another? Was every possible combination possible? And so he did the experiment where he took round yellow seeds and bred them with green wrinkled seeds. Round is dominant to wrinkled, and yellow is dominant to green. And so he did this experiment, and the results that he got were like this. If all the recessives had to go together and all the dominants had to go together, then your only possible combination of gametes here would be big R, big Y, and little r, little y. Those would be your only possible combinations. And so that's what you have here in the Punnett square put together. And these would be your results. You would get 3 yellow round and 1 wrinkled green because the recessive traits would always be traveling together. But what he actually got were 16 different combinations, or 16 different offspring, 9 out of 16 of which are round and yellow, 3 of which were green and round, 3 of which were yellow and wrinkled, and 1 out of 16 were green and wrinkled. And that can only happen if these assort independently, which means that you basically do FOIL to see all the combinations. The big R can go with the big Y, the big R can go with the little little R can go with the big Y, and the little r can go with the little y. So you get all 4 of those combinations that we see here, right? Note that each gamete only has 1 allele of each trait, right? That's from the previous experiment where we said that the alleles separate. 1 allele from each trait goes into an egg or a sperm. So it has 1 R and 1 Y. 1 R, 1 Y. That's true of all multiple trait Punnett squares that we will ever do. They get 1 allele for each trait. So 1 R and 1 Y. And then we put those together. So that's 1 egg that, or 1 sperm that this plant could have made. This is 1 egg that it could make. Then we combine the eggs and the sperm in the Punnett square here to see what our possible offspring would be like. And they now have 2 R's and 2 Y's, 2 of each trait. And so that's how we do the multiple trait Punnett squares. And these results of Mendel's proved independent assortment, which means that it's not that all the dominants move together and all the recessives moves together. They assort completely randomly, and all possible combinations are available in the offspring. And so that's how you get new combinations. If they assorted independently, then all of the offspring would look exactly like one of the parents. So you had round yellow seeds or wrinkled green seeds. So your offspring can only be those 2 combinations. But when you have independent assortment, you can get new combinations of genes. Here we have yellow wrinkled seeds. There was no yellowed wrinkled seed in the parent in this parent generation. That's a new combination of traits. Here we have round green seeds. We didn't have any round green seeds up here. So it's a brand new combination of traits. And when Mendel actually did this experiment, that's what he got. And the only way that it's possible is through independent assortment.